Lysine is another amino acid which is produced on large scale. It is one of the essential amino acids and required for the nutrition of humans and animals. So this is basically useful to improve the nutritional quality of food. Lysine production has been described by one stage as well as two stage fermentation process, that is direct fermentation and dual fermentation. Production of lysine by direct fermentation, that is the one stage process. Kinoshita and his co-workers, they have described the production of lysine by using a homoserine auxotrope of Corynebacterium glutamicum bacteria. There are certain other bacteria like Brevibacterium flavum, species of Brevibacterium, then Bacillus licheniformis are the major producers. Mutants of Bacillus licheniformis, which were resistant to analog and homoserine auxotropes, are basically useful. These mutants produced approximately 30 milligrams per ml of lysine in the fermentation broth. Let us go back and check out the biochemical pathway for L-lysine. So it belongs to aspartate family. So as we know that aspartate is converted to aspartyl phosphate through aspartokinase enzyme. Then this aspartyl phosphate is converted to aspartyl semialdehyde. From this there is a branch point. It goes to dihydrodepicolinate and through a series of intermediates gets converted to lysine. Aspartyl semialdehyde then gets converted to homoserine through homoserine dehydrogenase enzyme. Then there is a branch point again here at homoserine. Homoserine through certain intermediates get converted to methionine. Homoserine gets converted to homoserine phosphate, and threonine further gets converted to alpha ketobutyric acid, and finally there is formation of isoleucine. Now the part that we need to understand in this biochemical pathway for the production of lysine is that the biocontrol or the control mechanism, feedback control mechanism. So you can see that aspartokinase enzyme is sensitive to the levels of two metabolites, lysine and threonine. So when the production of lysine and threonine is in excess, in combination they will go and inhibit the conversion of aspartate to aspartyl phosphate. So they will inhibit this aspartokinase enzyme. Other mechanisms are threonine is independently responsible for the inhibition of homoserine dehydrogenase enzyme. Methionine is again independently responsible for inhibition of homoserine dehydrogenase. So this is the biochemical pathway, and this will be helpful for us to understand the overproduction of lysine. This part we have already studied in last semester under strain improvement chapter. Now fermentation using Corynebacterium glutamicum. The fermentation medium consists of cane molasses. So the sugar concentration is approximately up to 20%. Then 1.8% of soybean meal as a nitrogen source. The other ingredients are also added. Optimum concentration of homoserine or threonine and methionine to support the growth and lysine production. Here why this is important, because if you see in this diagram, so there is use of homoserine auxotrope of Corynebacterium glutamicum. What is this homoserine auxotrope? It is this. So this mutant is not able to synthesize homoserine dehydrogenase. So this pathway is blocked. So this mutant, it has this pathway only. This pathway is blocked. So therefore, In order to grow this mutant, there has to be addition of homoserine in the medium, or there can be addition of methionine and threonine. Of course, this concentration should not be sufficient enough to cause inhibition of aspartokinase. The production starts approximately within 10 hours after inoculation, and it continues till 50 to 60 hours. The sugar's concentration rapidly decreases following 10 hours onwards due to consumption of sugar by microbes. Foaming of aerated culture can be controlled by addition of proper antifoam agents. The microbe approximately produces 44 grams of lysine per liter of. This is the course of lysine production. You can see the concentration of sugar, it decreases, and there is increase in the production of L-lysine. This is the sugar concentration, and this is the growth curve. This is the flow sheet for lysine production. Cornybacterium glutamicum added to the production medium in fermenter. The concentration of biotin is higher, that is 30 micrograms per ml. So this is an intracellular metabolite. Then cells are separated, lysed. Then the broth is exposed to, for after primary separations like filtration and centrifugation, the broth is exposed to ion exchange chromatography, followed by that crystallization, freeze drying, and cold storage. Another way of production of lysine is through dual fermentation. This is very important. It is two-stage process or indirect fermentation. So most of the times it is one of the way of commercial production of lysine. So in this production in two stages, first diaminopimelic acid, that is DAP, is first produced and then it is converted to L-lysine. So the first step is production of DAP and second step is production of lysine. So DAP is produced by a number of microorganisms. So a relatively large amount of acid are produced by some lysine requiring auxotropes of E. coli. So in the first step there is use of lysine auxotropes of E. coli. So this lysine auxotrope does not synthesize lysine and requires lysine in the medium. So due to deficiency of DAP decarboxylase required for the conversion of DAP to lysine. So this E. coli does not have DAP decarboxylase enzyme and therefore it is auxotropic for lysine. Therefore, this auxotropic E. coli will form only DAP and not lysine because this enzyme is missing, because this is an auxotrope. So DAP is produced by submerged fermentation method by using auxotropic E. coli. The production medium mainly contains glycerol, Constip liquor, and other ingredients. The inoculum for production medium is prepared by growing auxotropic E. coli for 15 to 20 hours at 28 degrees Celsius with aeration and agitation. This is submerged fermentation with aeration and agitation. Optimum yield through this is 9 milligrams per ml within 3 days. So this DAP decarboxylase enzyme that converts DAP to lysine is produced by a number of bacteria, and these bacteria are then further used in second half of fermentation. So in first half of fermentation, bacteria like auxotropic E. coli is used, which can synthesize only DAP, and in second step the microorganisms are used which can convert DAP to lysine. Such bacteria are Aerobacter aerogenes and Prototropic E. coli. So these bacteria are prototropic and they can synthesize, they contain DAP decarboxylase enzyme. Uh, Another important property of these bacteria is that there is absence of lysine decarboxylase enzyme in them, because lysine decarboxylase enzyme is required for conversion of lysine to cadaverine. So that is another property, that these bacteria do not have this lysine decarboxylase. Then after propagation, the cells are separated from the medium by centrifugation and used directly or freeze dried as and when required. So for example, the cells from 25 ml growth medium were added to 300 ml of Erlenmeyer flask, which contained 100 ml of broth with DAP. So this is broth with DAP, and to which the cells of Aerobacter aerogenes are added. Then this broth is allowed to react or mix at pH 7.2. Then there is addition of 5 ml of toluene. Now why toluene is required? Because Aerobacter aerogenes has this DAP decarboxylase enzyme which is required for synthesis of lysine, and it is intracellular. So in order to release this intracellular enzyme extracellularly, cell lysis is required, and for which there is addition of toluene. So it lyses the cells and it liberates enzyme. Then the flask is incubated under shaking conditions so that the enzyme should decarboxylase enzyme should come in contact with DAP, so that there will be conversion of DAP to lysine. This is the biochemical reaction. This is DAP, diamino pimelic acid. So DAP decarboxylase enzyme converts this to lysine through a release of one CO2 molecule. It is decarboxylase, so one molecule of CO2 is released. Other ways of lysing the bacterial cells are butanol or ultrasonic vibrations. After conversion, this reaction mixture is filtered, then adsorption chromatography is used, then further purification is done by cation exchange resins such as Amberlite R120. Then the elution is done and further processings are done.